So Bepranemab targets the mid-region of tau. It's an anti-tau antibody, and the region it targets is proximal to the microtubule binding domain, which is an area that's essential for tau accumulation. We have preclinical work that's shown that Bepranemab has the potential to prevent the induction of tau pathology and block its spread through the brain. And we believe it's doing this by or through binding of extracellular tau aggregates that are released from neighboring cells in a process known as seeding, and that's where we believe the pathology originates from, is this seeding or essentially a downstream infection type process. Obviously, amyloid has garnered a lot of interest. By what we have observed or what's been reported in the literature is that the clinical effects of Alzheimer's disease are more correlated with tau load. And so the idea is that the sooner we can act and prevent the onset of the clinical manifestations, the better off. By the time, or if we look at later on in disease, it may be too late to make a material impact. So in this case, at least the initial hypothesis here is prevention is really our target of interest. Bepranemab has been tested in 2 healthy volunteer trials, one healthy Caucasians and one in healthy Japanese. The drug has been safe and well tolerated in both trials. The reported AEs were similar across all volunteers between Bepranemab and placebo. Bepranemab has also been tested in the parasupernuclear palsy patient population, a small phase 1b trial with 25 patients. Most of those patients have transitioned into an open label extension. The drug has been well tolerated in the PSP population as well. And now we're evaluating next steps. The trial itself is a global multicenter patient and investigator blind trial, placebo-controlled parallel group, and we're investigating the efficacy, safety, and tolerability of IV administered Bepranemab versus placebo in the early AD population. It's the population split is approximately 40% prodromal AD and 60% mild AD patients. 450 or approximately 450 patients are intended to be enrolled across approximately 125 centers in 10 countries. Randomization is 1:1:1 to receive placebo, low-dose Bepranemab, or high-dose Bepranemab via infusion every 4 weeks for 76 weeks. And the primary endpoint will be read out at week 80. The primary endpoint is the CDR, is change from baseline in the CDR sum of boxes. At the completion of the double-blind period, participants will be eligible to enter into an open-label extension period, and this is planned to be low-dose or high-dose Bepranemab every 4 weeks for 48 weeks. The trial has been ongoing since June of 21, and we have just recently activated our final centers. As of June 1st, which was, I believe, the cutoff for the poster, we had enrolled 1,079 participants. 696 have failed screening, and 172 have been randomized. The main reasons for screen failure included not meeting the amyloid beta positivity requirements, so either by CSF assay or amyloid PET, or MMSE entry criteria, which is greater than or equal to 20. We think that targeting extracellular tau will have an impact on the clinical manifestations, and we're hopeful that downstream will prevent downstream seeding. As I mentioned, we do have preclinical data that suggests bepranemab does prevent seeding, at least at an in vitro level. And so we are hopeful that that'll translate into the clinic. At UCB, we're focused on solving some of the greatest challenges in neuroscience, including developing treatments to prevent, delay, or stop disease progression of neurodegenerative disorders such as Alzheimer's and Parkinson's disease. In AD, we're collaborating with Roche to develop a disease-modifying therapy, which is bepranemab. Our research is focused on the development of this unique antibody, which was discovered by UCB, and through which we aim to establish a new approach to treatment that may positively impact millions of people affected with AD.